Hi everyone. In this video, we'll be demonstrating two different ELISA tests related to SARS-CoV-2, the virus that causes COVID-19. The first is an antigen detection ELISA, which is used to diagnose an active infection, and the second is an antibody detection ELISA, which measures a person's immune response or past infection. Both of these tests rely on a specific interaction between antigens and antibodies, along with an enzyme that creates a visible color change when the reaction occurs. Throughout the demonstration, we'll be showing each step using our handmade model pieces and answering the guiding questions along the way.
ELISA. In this type of ELISA, we test a patient sample for the presence of viral antigens such as SARS-CoV-2 spike protein. This is called a sandwich ELISA because the antigen becomes trapped between two layers of antibodies, a primary antibody and a second enzyme-linked antibody. When the enzyme reacts with the substrate, the sample will change color to present a positive test result, meaning the viral antigen, like SARS-CoV-2 spike protein, is present in the sample. First, I get the patient's swab sample. Then I begin by coating the ELISA well with the primary antibody. This antibody is designed to capture a specific viral antigen. After incubation period, I wash the well to remove any unbound antibodies, leaving a layer of primary antibodies firmly attached to the surface. Next, I add the patient's sample to the well. If the virus is present, its antigens will bind specifically to the immobilized primary antibodies. After a short incubation period, I wash the well again to remove unbound proteins and debris. Then I add the enzyme linked secondary antibody. After several wash steps, only the properly bound antibody antigen antibody sandwich complex remains. Finally, I add the substrate. If the antigen was present in the patient sample, the enzyme on the secondary antibody catalyzes a reaction that turns the well blue. A positive result, increasing in color intensity, correlates to the antigen amount. No antigen was present, the enzyme has nothing to react with, and the well remains clear, a negative result.
Now let's talk about an important part of every ELISA, which is in the controls. For the positive control, we'd add a sample that contains a known amount of the viral antigen. This well should definitely turn blue, which confirms that all of the reagents and steps in the test are working correctly. For the negative control, we'd use a sample that does not contain the antigen. This well should stay clear, showing that there's no contamination or nonspecific binding happening in the assay. Together, these controls help make sure the test results are accurate and reliable. If we tried using this antigen detection ELISA 35 days after infection, it probably wouldn't give a reliable result. By that time, the immune system has already cleared most of the virus, so the amount of viral antigen in the body would be very low or even undetectable. That's why this type of test is really only useful early in the infection when viral antigens are still present in high enough concentrations to be detected. PCR testing, which looks for viral RNA rather than proteins, also most useful early in the infection, during the same period when antigen levels are high. Later on, even after the virus has been cleared, PCR can sometimes still detect leftover fragments of viral RNA. But by that point, an antigen ELISA would already be negative because there are no more viral proteins present. Here's a good question about antibody pairing. Could the same goat anti-rabbit secondary antibody detect different rabbit antibodies, for example, one against influenza and another against HIV? The answer is yes. That's because the goat anti-rabbit antibody binds to the constant region of rabbit antibodies, a region that's the same regardless of what antigen the rabbit antibody is specific for. So it doesn't matter which antigen the rabbit antibody targets, as long as it's a rabbit antibody. The same goat anti-rabbit secondary will detect it. If we attach the enzyme directly to the primary antibody, that's called a direct ELISA. We'd still see a color change when the substrate is added, but the signal would be weaker because each antigen would only have one enzyme molecule attached. In contrast, when we use an enzyme linked secondary antibody, like we're doing in this indirect ELISA, the signal gets amplified. That's because multiple secondary antibodies can bind to a single primary antibody, bringing in many more enzyme molecules. The big advantage of the indirect method is a stronger color signal and greater sensitivity, which makes it easier to detect even small amounts of antigen. So to summarize, the antigen detection ELISA works by directly identifying viral proteins in a patient sample. This tells us whether the person is currently infected with the virus. Next, we'll move on to the antibody detection.
Now we will talk about the antibody detection ELISA. This second assay is called an indirect ELISA, and it is used to detect antibodies in a patient's serum that recognizes SARS-CoV-2 proteins such as the spike glycoprotein. A positive result here means that the patient's immune system has responded to the virus, either through infection or vaccination. The first step is different from the antigen detection ELISA. Instead of adding the patient sample, the well is coated with a purified viral antigen, such as the SARS-CoV-2 spike protein. This known antigen will later capture any matching antibodies from the patient's serum. In step 2, we add a wash buffer. After coating, we wash the wells to remove any unbound antigen. Washing is important for 2 reasons. First, to remove unbound materials, and second, to prevent nonspecific binding that could cause a false positive result. In step 3, we add the patient's serum. If the patient has antibodies against the coronavirus spike protein, those antibodies will bind to the antigen on the plate. If the patient has never been exposed or vaccinated, nothing will bind and the well will remain empty after washing. In step 4, the unbound serum and antibodies are washed away. Next, the enzyme-linked secondary antibody, such as GO antihuman IgG, which binds to human antibodies, is added. If the patient's antibodies are present, the secondary antibody attaches and binds the enzyme with it. Unbound secondary antibody is then washed away. In step 5, the substrate is added. If the patient's serum contained anti-SARS-CoV-2 antibodies, the enzyme reacts and turns the solution blue, a positive result. However, this doesn't necessarily mean the person currently has COVID-19. It could simply mean that they were infected in the past or have been vaccinated, because antibodies persist long after the virus is gone. Here you can see a positive result that has reacted and has turned blue, and a negative result, no reaction has occurred, and the serum remains yellow.
Just like before, we need to run several controls to make sure the antibody detection ELISA is working properly. First, we have a positive control using a serum sample that's known to contain anti-SARS-CoV-2 antibodies. This well should turn blue, confirming that the reagents and procedures are working. Next, we have a negative control using serum from someone who has never exposed to the virus. This well should stay clear, showing there's no background signal. Finally, we include a negative patient sample, which would also remain clear if that patient never produced antibodies. If we tested someone 35 days after exposure using the antibody ELISA, the answer is yes, it would work. By that time, antibody levels are high and stable, which makes the antibody ELISA much more accurate for late diagnosis or more monitoring immune responses compared to the antigen test. Both the antigen and antibody ELISAs rely on a specific interaction between antigens and antibodies and on an enzyme reaction that creates a visible color change. Together, these tests give scientists and healthcare workers the tools to figure out who is currently infected, who has recovered, and who has developed immunity. Thank you for watching.
Detection ELISA.